Hello students, in this video we are going to see about the next topic in developmental biology, that is cell-cell interaction. Cell-cell interaction. This is the today's topic. In cell-cell interaction, we have two broad classifications. First one is proximate or inductive interaction. Proximate or inductive. Proximate or inductive interaction. Cell-cell interaction is nothing but, as the name suggests, this explains about how two cells in our body interact by passing some signals like paracrine signals, by using hormones, by using secondary messengers. Two cells generally interact. So we will see all the methods in which the cell generally interact with respect to the perspective of developmental biology. This is first type, proximate inductive interaction. Second type is epithelial mesenchymal interaction. Epithelial and mesenchymal interaction. Epithelial mesenchymal interaction. These are the two types. First type is proximate or inductive interaction. Second type is epithelial or mesenchymal interaction. These are the two types that we are going to learn about in this video. First we will see about proximate or inductive interaction. Inductive interaction. It itself is of two types, that is instructive and permissive. Epithelial mesenchymal interaction, EMI, epithelial mesenchymal interaction is of two types. One is regional specificity of induction. Regional specificity of induction. Second thing is genetic specificity of induction. Genetic specificity of induction. So broadly summing up all the classifications we have studied, cell-cell interaction is of two types, proximate or inductive. Second type is that EMI, that is epithelial mesenchymal interaction. This is classified into two types. This is classified into two different types. Instructive interaction and permissive interaction. Regional specificity of induction and genetic specificity of induction. Proximal and inductive signal is again proximal and inductive cell-cell interaction is classified of two types, that is instructive interaction and permissive interaction. Epithelial mesenchymal interaction is divided into two types, that is regional specificity of induction and genetic specificity of induction. In this video we will learn about this instructive interaction and permissive interaction and their differences among these two classifications. First is instructive interaction. In instructive interaction, as the name suggests, some cell or something else is giving instruction to the target cell. This is the general meaning. General meaning of instruction is instructing somebody, instructing something to do some work. Something is instructing our target cell to achieve the goal. It can be hormone, it can be secondary messengers, it can be paracrine signaling. So this is inducer. Our target cell is receiver. Okay. This inducer will send signal to the receiver. Receptor of the receiver will accept the signal from the inducer, and it will act accordingly to achieve the goal. Here we have two players, that is inducer and receiver. Receiver will be our target cell whom we want to do specific work. Inducer will give signals to the receiver and the receiver will act accordingly. Okay. Then another terminology we have already learnt is competence. This target cell should be competent to receive signal from the target cell should be competent to receive the signals from inducer. Only then the target cell can react. Okay. What is competent? What is competence? Competence is nothing but ability to do work. Competence is nothing but ability to do work. Inducer will produce signals. The signal has to be received by the receiver. To receive this signal, our target cell should be competent to receive the signal. The target cell should be competent to process the signal. Only if it accepts and processes the signal, it can give the desired output. So the competent means the ability of the cell to do work. That is competence. This is instructive interaction. Summing up, instructive interaction, receiver, we have an instructor, we have a receiver, and we have a success, a work done by the receiver. This is inducer. This is receiver. Inducer will send signals to receiver. Okay. Receiver will accept those signals. It will have a specific receptor to accept the signal. Inducer will accept the signal, will send the signal, and receiver should be competent to accept the signal. After accepting the signal, the receiver process and work accordingly. result of ability. Hope everyone understood the meaning of instructive interaction. Next, we are going to see about permissive interaction. Permissive interaction. Permissive interaction is nothing but a signals from environment. Signals from environment. So let's see what is the meaning of this. So let's have a petri dish. And we have few cells. And we have few signals in the environment. These cells will respond to the signal from the environment. So here the signals are not given by the inducer, rather the signals are given by the environment. This is the meaning of permissive interaction. Here the signals are from the environment. This can be done in vivo, this can be seen naturally in very conditions of in vivo conditions. This can be done in various in vitro standard lab conditions as well. In case of stem cell, if we are growing, consider this in a petri dish, and if we are growing so many stem cells in it along with various signals from the environment. Consider this green ink as signals from environment. These signals from environment of this petri dish will help the cells to grow according to our desired fate. Okay. So here the signals are from the environment. This is called as permissive interaction. Will be helpful for anyone from life science background who are maintaining cell lines. This will be done in other unit as well when you study about cell lines. This is called permissive interaction. In permissive interaction also we have few things, that is signals. Signals can be chemical messengers, hormones, secondary metabolites, any substances. Signal will attack our target cell and it will do work and it will attain its fate accordingly. So this is the basic idea of permissive interaction. No two cells are involved. No two cells are involved. It is only about the interaction between no two cell involved. It is only interaction between environment and target cell. Hope In this video, I have made clear the concepts of instructive and permissive interaction. In instructive interaction, inducer will give signal to receiver and the receiver is so competent and it attained the specified cell fate. And it attained its specified cell fate. This is called instructive interaction. In permissive interaction, we have signals from environment that help the cells of desired cell and it give rise to desired fate. These signals from environment are so specific to our target cell so that it interact with the receptors in the target cell. Hope these concepts of instructive and permissive interactions are clear. In this video, we have seen about instructive interaction and permissive interaction. It comes under the topic of proximate or inductive interaction under the heading of cell-cell interaction.